discussing about the enhancement of rate of reaction by enzymes. So enzymes enhance the rate of reaction, means they do biocatalysis. Now in this video we will learn about how exactly these enzymes are capable of catalyzing the reactions. Okay, so generally the enzymes are able to enhance the rate of reactions. They enhance the rate of reactions by lowering of activation energy. Lowering of activation energy. This is the main mechanism by which the enzymes are able to catalyze the reactions, catalyze the conversion of substrates to products. They lower the activation energy. Before we understand more about lowering of activation energy, first let's try to understand what exactly is activation energy. We shall look at this graph to have a good understanding about activation energy. So this is a graph plotted between reaction coordinate taken on the x-axis and free energy taken on the y-axis. In this graph, you can see the substrates are here. I mean the energy levels of substrates are here and the energy levels of products are here. But you can see a bump in the graph, that is whenever the substrates have to be converted into products, they have to move to a state called as transition state, which is represented by T. Okay. For the substrate molecules to move into transition state, they require some amount of energy. They have to increase their energy levels. They require some energy so that they move from the substrate energy level to the transition state. This energy that is required for the substrate molecules to move to transition state, so you can see here, this is the amount of energy, the energy gap or the energy required for the substrates to move into transition state can be defined as activation energy. This is activation energy. Okay, the pink line represents the reaction, I mean the conversion of substrates into products in the absence of enzyme. Okay, it is a non-enzymic reaction. So substrates getting converted to products in the absence of enzyme is represented with the pink line. Means you have to understand that this much amount of energy is required for the substrates to move to transition state. Once they move into transition state, they will be next converted into products. Now what exactly happens in case of an enzyme catalyzed reaction is that in an enzyme catalyzed reaction, a lowering of activation energy will occur, which means you can look at this yellow colored line which represents the energy changes in case of an enzyme catalyzed reaction. So here if you see, substrate's energy level is here and the transition state energy level is here, means this much of energy is required for substrate molecules to get into transition state. So this here, the white line represents the activation energy in absence of enzyme and the yellow line represents activation energy, that is energy required for substrate molecules to get into transition state in the presence of enzyme. With enzyme means very less amount of energy is required whenever enzyme is present and very high amount of energy is required. The activation energy is very high whenever enzyme is not there, is not catalyzing the reaction. Now we shall see exactly how this enzyme is able to lower the activation energy. Yes. So if this is the structure of your enzyme, So while discussing about properties of enzymes, I told you that enzymes will be folded in such a way that the activation site will have certain amino acids, or it will have the functional groups of certain amino acids which are required for binding of the substrate molecule. So this is an enzyme and this is the active site of the enzyme. The active site of the enzyme will be lined by the functional groups of the amino acids that are required for binding with your substrate molecule. So the substrate molecule can bind like this with the active site of the enzyme. So this you call it as enzyme substrate complex. Binding of the substrate to the enzyme will produce something called as enzyme substrate complex. This is an enzyme substrate complex. Once the enzyme substrate complex is formed, what happens is the substrate is going to interact with the functional groups present in the active site. These are weak interactions and these are non-covalent interactions. Such interactions between the substrate and the amino acids present in the active site of the enzyme will result in production of small amounts of free energy. I repeat, the interaction between the substrate molecule and the amino acids that are present in the active site of the enzyme will result in the production of small amounts of free energy, okay, which is referred as binding energy. This is called as binding energy. So this binding energy released by the interaction between substrate and the enzyme active site is going to contribute for the activation energy for an reaction. That's why in case of enzyme catalysis reactions, the activation energy will be lowered. To make this concept little simple, I will give you an example. Say for example, you want to buy a book worth 100 rupees. So as to buy a book worth 100 rupees, you have to earn 100 rupees. Okay, this is something like when you want to convert substrate, and when you want to move your substrate to the transition state, you require some amount of activation energy. Say for example, it is 100 rupees. Okay. If your father does not support you, you yourself have to earn all the 100 rupees so as to buy the book. Then the amount you have to earn will be a bigger one. I mean the total 100 rupees. For example, if your father contributes 50 rupees for your book, then you need to earn only another 50 rupees. This is something like this. The enzyme interaction with the substrate is contributing for release of some free energy, which we call it as binding energy. When this interaction is contributing for the release of binding energy, obviously the total amount of activation energy that you require, required for the reaction to proceed will get lowered. In this way, the activation energy will be lowered in case of an enzyme catalyzed reaction.